Proteins are the most diverse group of all the macromolecules. They have many different functions. Defense proteins protect our body against invaders. Enzymes and hormones elicit a cellular response and communication between cells. Examples of structural support include keratin in our hair, collagen in our skin, and silk fibers used by spiders. Hemoglobin is a protein that transports oxygen. Contraction of muscles is due to actin and myosin proteins. And the movement of sperm is due to a motor protein. And lastly, albumin, or egg whites, provides nourishment to developing embryos. Proteins are polymers made up of amino acids. So know that the monomer, or the single unit, is an amino acid. Proteins are very large and complex and have a 3D shape. We refer to the polymer as a polypeptide. The term polypeptide simply means made up of many peptides. One or more polypeptide chains can be folded and bonded together. This is what leads to the very large three-dimensional structures. There are 20 amino acids. All proteins are made up of different combinations of these 20 amino acids. Each amino acid has a similar structure template. Each has a left side chain called an amino group. That's the nitrogen with the 2 hydrogens. And a right side chain with a carboxyl group, the COOH. Notice that this also shows the amine group on the left, the carboxyl group on the right. The central carbon atom has a hydrogen attached, and then an R group. The R group is what is different for each amino acid. Compare the amino acids here. The colored region of each amino acid is that R group. This is the only region that differs between each of them. You can also see that they are grouped into different classifications. There are nonpolar, polar, and then there are those that are electrically charged. These characteristics affect the shape of the protein. Recall that amino acids link together to form a peptide. Amino acids are linked by dehydration reaction. Look at the orange area at the top of the picture. The carboxyl group of one amino acid will bond to the amino group of another amino acid, forming a peptide bond and removing 2 hydrogen and an oxygen. Everyone knows that this forms water. Therefore, a dehydration reaction kicks off water. The result is the 2 amino acids bonded together. We call this a dipeptide. This process repeats until all amino acids have linked together. This is now a polypeptide because it contains many amino acids. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. I use this simple analogy to explain them. Can you imagine being able to write a paragraph if alphabets didn't exist? In fact, we have to go through a hierarchy of complexity before we can even attempt to write a paragraph. The alphabet is needed to construct words. Words are needed to construct sentences, and sentences are needed to construct a paragraph. Similarly, a fully functional protein is assembled through these 4 levels of hierarchy, as illustrated next. The primary structure at the top are the letters. The secondary structure are the words, the tertiary, the sentences, and quaternary, the paragraph. Each of the 20 amino acids have their own abbreviations. The abbreviations are used to list the amino acid in the order they appear in the protein sequence. This is the primary structure. The secondary structure includes the folds and coils that proteins can have. These are known as alpha helices and beta pleats. These occur because of the hydrogen bonding that happens between the backbone of the amino acids. tertiary structure is determined by the interactions between R groups rather than the backbone. Interactions include actual ionic and covalent bonds called disulfide bridges, which help reinforce the protein structure. Hydrophobic and hydrophilic regions may also affect the protein structure. The tertiary structure is what gives rise to people with curly hair. The more disulfide bridges you have, the curlier your hair will be. The quaternary structure results when two or more polypeptide chains form one molecule. Collagen on the right is a protein picture. It is a fibrous protein consisting of 3 polypeptides coiled like a rope. The shape of each protein is unique. Proteins do their job because of their shape. A slight change in primary structure can affect a protein structure and ability to function. Sickle cell disease, an inherited blood disorder, results from a single amino acid substitution in the protein hemoglobin. Normal hemoglobin is pictured on the left and sickle cell on the right. Notice the primary structure for each. A change in the protein sequence occurs in the 6th amino acid, highlighted in pink. Because of this change, the secondary and tertiary structures are significantly different in each. This slight difference has caused the shape to change to a sickled molecule, which then caused the function of that protein to change. These sickled molecules then stick together, decreasing their ability to carry oxygen. Physical and chemical conditions can affect structure. pH, salt concentration, temperature, and other environmental factors can cause a protein to unravel. Denaturization is the loss of a protein structure. Denatured proteins are biologically inactive. People with curly hair can use a straightener to straighten their hair. This heat denatures the protein or breaks the disulfide bridges between the proteins. Enzymes are a type of protein that acts as a catalyst, speeding up chemical reactions, some up to 1 billion times faster. Enzymes end in ACE, A-S-E. Recall what sugars end in, O-S-E. Enzymes can perform their functions repeatedly, functioning as workhorses that carry out all of the processes of life. Enzymes are highly selective. They only accelerate chemical reactions for a specific molecule. 3 examples seen here are digestive enzymes. Protease speeds up the reaction or breakdown of proteins, lipase of lipids, and amylase sugar molecules made up of amylose. On the top left, we see the enzyme sucrase. The active site allows the substrate sucrose to fit perfectly into it. It will only allow sucrose to fit. This now speeds up the breakdown of this molecule that would not happen naturally or would take a very long time to. The end result is a glucose and fructose molecule that is released. That enzyme is then available for another reaction. Enzymes are never used up in the reaction. They simply help speed up and facilitate the reaction. Enzymes also end in ACE, match the molecule that ends in ose. All reactions require energy to occur. The amount of energy required is called the activation energy. In this energy diagram, you can see that the amount of energy required for the reaction to occur is significantly lowered, shown by the blue line, when an enzyme is present. Another term we use, again, is a catalyst, meaning to speed up. This picture is another way of thinking about enzymes. Enzymes speed up reactions by lowering the activation energy, represented by the rocks in these pictures. The rocks are much higher on the left where there is no enzyme present. Notice that with the enzyme on the right, the rocks are much lower. All the frogs will be able to jump over the barrier much quicker than the frogs on the left.